Hello friends, dear students. Today we are going to have a lecture on plague. Plague is a zoonotic disease and it is transmitted via the rat flea. This is the picture of the rat flea. Rat flea is a insect and it bites and transmits the plague bacilli through the bite. And usually it bites on the lower limbs because these rat fleas, they do not fly but they hop, and therefore they can reach, by hopping they can reach a distance of 1 to 2 feet, and that's why the lower limb is most affected. Now let's see the some historical aspect of plague. Plague is also called as the Black Death. Let's see why it is named as Black Death. Plague is primarily a disease of rodents and their fleas, which can infect humans. It is a zoonotic disease mainly affecting the rodents, seen in the rodents. Rodents are most commonly rats. It is transmitted between. So, plague is primarily a disease of rodents and their fleas, which can infect humans. It is transmitted between the rodents by the rodent fleas. So the fleas are the vectors and the host is the rodent. And it can be transmitted to people when the infected rodent fleas bite them. Primarily it is a zoonotic disease. Plague is a serious disease in people with a high fatality rate of 50 to 60% if left untreated. Plague has been responsible for widespread pandemics with high mortality. It was known as Black Death during the 14th century, causing an estimated 50 million deaths, approximately half of them in Asia and Africa, and the other half in Europe, where a quarter of the population succumbed. Plague has declined dramatically since the last century, in the last early part of 20th century. It is primarily because of the improvement in the living standards and health services. However, a substantial number of countries continue to be affected by plague and the case fatality rates remain high and antimicrobial resistance has begun. Therefore, continued vigilance is required, particularly in human populations living near the natural plague foci. Plague foci are not fixed. Okay, the focus can shift, and it can shift because of factors related to climate change, landscape, and the rodent population migration. Natural foci of plague are situated in all the continents except Australia, within the broad belt in tropical, subtropical, warmer, temperate climates. As you can see in this diagram, in the picture, you can find that all the countries are affected except for Australia. Plague is transmitted between rodents and other animals via the wild fleas. Wild plague exists in the natural foci independent of human population. So human beings are not the usual hosts, but they are accidental hosts. Usually the cycle is maintained between the fleas and the rodents. Domestic plague is intimately associated with rodents living with humans and can produce epidemics in both human and animal populations. Humans are extremely susceptible to plague and may be infected directly or indirectly. Indirect transmission through the bite of a flea is a common route of transmission between the plague infected rodents and fleas. So, direct transmission can occur human to human, which is not common, but indirect form is common through the bite of the flea. So it's now let's see. It's primarily a zoonotic disease. Okay, zoonotic disease means disease which is prevalent in the animals, in mammals, and transmitted to humans through such mammals. So here the rodent is involved as a animal. Pests involved are rats and the rat flees the vector. Zoonotic diseases are the agents of which are naturally transmitted between vertebrate animals and man. It exists in natural foci and transmitted by infected flea bites to human beings living in the same ecological and environmental condition. There are three different forms of plague: epizootic, enzootic, and sporadic. So whenever there is an epidemic among the rodents in the forest, it is the epizootic. It's also present endemically, and it can also occur in the sporadic format. Now before we discuss plague, let us see some of the flea-borne diseases. The rat flea acts as a vector in plague, in endemic and murine typhus, in sugar disease, in tularemia, and in the infection by Hymenolopis diminuta. So these are the plague-borne diseases. The mnemonic here is the bench, where B stands for bubonic plague, E stands for endemic typhus, CH for shigellosis, and H for the Hymenolopis infection. Now, what are the diseases spread by rodents? So, rodents are responsible for spreading bacterial diseases like plague, tularemia, salmonellosis, viral diseases like Lassa fever, hemorrhagic fever, encephalitis, rickettsial diseases like scrub typhus, murine typhus, rickettsial pox, then parasitic infections like Hymenolopis diminuta, leishmaniasis, amoebiasis, trichinosis, and other diseases like rat bite fever, leptospira, histoplasma, etc. Now, what is the definition of a case of a plague? Disease characterized by fever, chills, headache, severe malaise, and prostration. Bubonic form of plague causes extremely painful swellings, especially the lymph nodes of the inguinal region, since it bites on the lower limb and the drainage is to the inguinal lymph nodes. The buboes are formed in the inguinal region. Then we have the pneumonic form, which causes cough, blood-stained sputum, chest pain, and difficulty in breathing, and septicemia. So there are three main types: bubonic plague, pneumonic plague, and septicemic plague. So as you see in the history, it has occurred in the 542 BC, and it is called commonly known as a Justinian plague, where it led to the deaths of around 100 million people. And there have been many such pandemics of plague. Then again in the 1346 AD, it led to 25 million deaths. Again in 1894 AD, it was seen and it had established the foci in Asia, Iran, Africa. So again the third pandemic of plague. And after that, in 1950, 1980, 1938, we have seen cases of plague erupting, but the 20th century, the number of cases have been declining because of the overall improvement in the hygiene and the living standards of people. Now, what happened in 1994? In India, there was a return of plague. 4 lab-confirmed human bubonic cases of plague were found or reported in the Beed district of Maharashtra, and an outbreak of pneumonic plague in Surat, followed by a series of reporting of cases from various parts of the country like Delhi, Mumbai, Kolkata, and some other places. 4,780 cases were suspected, out of which 167 tested positive and 53 patients died. Again, in the year 2002, 16 pneumonic plague confirmed cases were found, with 4 deaths in the city of Simla. So this is the history where the plague for a long time was thought to be a disease which had disappeared, but it made its reappearance in 2002. The plague was controlled in the 20th century because as a byproduct of the malaria control program. Only 6 cases were seen in 1966, and after that a reappearance in 1993 in the Beed district of Maharashtra after the earthquake, in Surat after a heavy flood in the river Tapti, and in Himachal Pradesh. The zoonotic foci continue to persist in the Kolar and the Bangalore district of Karnataka, Dharampur in Tamil Nadu, Chittoor in Andhra Pradesh, and also in Himachal Pradesh. Now let's see the epidemiology. Epidemiological factors. First discuss the agent factor. Yersinia pestis is a gram-negative, non-motile cocobacilli. It is stained using the Wason stain, and on staining it shows the bipolar staining. Blue colored bipolar bacilli can be seen. It is found in abundance in the buboes, blood, spleen, liver, and the other viscera. It produces the endotoxin as well as the exotoxin, and many other antigens are present. And it can survive and multiply in any favorable climatic conditions and even in the soils of the rat burrows. So it can live even outside the body of its host or vector. The natural reservoir: out of 1700 known rodent species, 200 are associated with plague. So the plague, this bacilli, they sustain in the rodent flea cycle, and the rodents act as a natural reservoirs. So the wild rodents, field mice, gerbils, skunks, other small animals, Tatara indica, which is the commonest in India, that is the wild rat, and he is the most common reservoir. While when the plague spreads among the human population, the rodent involved is the Rattus rattus. And in the peridomestic transmission, it is the Rattus nervogus. The source of infection. All of you should know the difference between source and reservoir. Reservoir is the human or the animal or the natural habitat where the virus or the bacteria not only survives, it also multiplies. While source of infection is the human or the animal or any material like food, water, anything from which we acquire the infection. So in the case of plague, the source of infection is the infected rodent, flea, and it can also be the case of pneumonia in case of pneumonic plague. Now let's see the host factors. Age and sex. All the ages and both the sexes are susceptible. Human activities which offer numerous opportunities of the man-flea-man contact. So whenever the man or the human enters in the forest areas or enters in the natural foci of the rodents and the fleas, there is a chance that they may get infected after the bite of the flea. The movement of people is responsible. There is no natural immunity and a relative immunity acquired after getting infected. Sorry. Seasons. The plague season when the temperature and the humidity is around 20 to 25 degrees and 60% respectively. Rainfall. Post rainfall, the commonly epidemics were seen because the rains used to fill the rodent burrows and the rodents used to come out from the burrows and spread amongst the on the surface. More of the urban rural phenomena, untoward ecological conditions and lack of efficient vector flea control measures. Now let's talk about, we have discussed the host factors and the agent factors. Now who is the vector for plague? The plague vector is the Xenopsiala chyopis, is the commonest and the most efficient vector. So the rat flea, what we call, its scientific name is Xenopsiala chyopis. The other rat fleas are Xenopsiala astia and Xenopsiala brasiliensis. Rat flea, uh, Nasophilus fasciatus and rat flea Pulex irritans. Both the sexes are involved, unlike mosquito where only the female bites. Now we come to an important term, what is a blocked flea? Fleas which have ingested bacilli whose containing the proventricular is blocked after repeated multiplication of bacilli and hence the flea cannot take the food. So the rat flea, while sucking blood, it also ingests the plague bacilli and these bacilli then multiply in its proventriculars and completely block those proventriculars and then she is not able to take her food. The partially blocked flea is more dangerous than the completely blocked flea because a completely blocked flea will eventually die of starvation, but partially blocked flea will keep on hitting and biting others and transmitting the infection. Now the flea indices is a Schiopis index, the total flea index, incidence of flea species, burrow index. These do not indicate plague epidemic but definitely act as a warning sign, and we also suggest that we need more stringent control measures are needed to protect the human population. Modes of transmission. So how does plague spread? It can spread through the bite of the rat flea. It can spread through the mechanical transmission of the bacteria through the flea, or it can spread through its feces. Now plague in the rodents, we can see them under epizootic format where it is present in the natural foci and suddenly there is a fast multiplication of the bacilli and affection of large number of rodents. So that is the epizootic period. After the deaths of many of the rodents, suddenly the number of affected rodents come down drastically and those are called as the silent periods. At that time, the transmission of the bacilli remain dormant in the burrow soils, and again when the conditions are favorable, the rat fleas are infected, and they also spread it back to the rodents. So this is the cycle which happens in the science which deals with plague in animals is epizootology, and the plague exists in two ecological cycles, the wild plague and the domestic plague. Wild plague exists in nature, independent of human population and their activities, while domestic plague is associated with man and rodents living in and around the human population. Natural foci. So I have been using this term. What is its meaning? A strictly delimited area where ecological conditions ensure the persistence of the etiological agent for considerable periods of time, and where epizootic and period of quiescence alternate without the introduction of infection from outside. Why plague persists for a long time in the natural foci? Where there is a rat-flea-rat cycle, then latent infections in the rodents. So the cycle or the bacilli are maintained in this rodent-flea-rodent cycle, or in the form of latent infection among the rodents, or the development of resistance to plague infection by some rodents with localization of infection in certain organs. So the bacilli get located in one organ like lung, and they remain there dormantly, just like the TB bacilli remains dormant in the humans. Then survival rate of the rat flea is as long as 4 years in rat burrows under optimal microclimatic conditions. Variations in the pathogenicity of Yersinia pestis. Survival and multiplication of Yersinia pestis in the rat burrow. Now the epidemicity of plague. Epizootic and enzootic cycle in each natural plague foci has specific cycle and periodic patterns. Infection from one bureau to another bureau is through the field rodents, infecting peridomestic rats. There are the licensed rodents, and then the domestic or the village rats, that is the commensal rodents. Outbreak of human plague is followed after the outbreak in the rodent plague. So initially there would be rat fall, or there would be the rodents which are affected, and those which are killed by the many are dying because of the plague amongst the rodents. And as the rodent population is decreased or depleted, the rat fleas start biting the humans. So there is an outbreak among the humans. Silent periods. Usually there are long duration of silent periods of 10 years or more, followed by a sudden explosion of rat and human cases in the natural foci. There is a disappearance for a long time and then reintroduction from other areas which have infected rodents, infected fleas, or infected birds. Yersinia can also multiply in the soil in a suitable microclimatic conditions. Rodents get reinfected through reoccupation of the infected burrow or the soil. So this is how the infection persists, because the bacilli is able to sustain itself in the soil, especially in the burrow soil, and the rodents can get reinfected when they reoccupy these infected burrows. Investigation. Collection and forwarding of dead rats, autopsy and collection of the smear, animal inoculation, pathological changes which are noted during the autopsy findings or the pathology. If you do of the lymph nodes, you find edema and necrosis at the site of injection, enlargement of the lymph nodes, enlargement, congestion and necrosis of the spleen, mottling of the liver tissue, necrosis of lungs, and congestion and hemorrhage of the subcutaneous tissues. So the rats have to be collected in the very aseptic precautions and in special the plastic containers, and they have to be sent to the lab, and the lab we do the smear examination of their if we collect the smears, and if we do the autopsy, findings are also there. Now the mode of transmission of human plague. It is generally the commensal rat when it bites the rats, and the rat flea will bite the commensal rat, and the same flea will again bite the human. So this is the rat flea man cycle. Then wild rodent flea and direct contact when they come to the man. Wild rodent, peridomestic rodent, commensal rodent, man, man human flea man, and again there is a man to man transmission. Now what is the incubation period? In the case of bubonic plague it is 2 to 7 days. Septicemic plague it is 2 to 7 days. And for the pneumonic plague it is 1 to 3 days. So pneumonic plague is a very acute infection and has a very short incubation period, and that's why it is very dangerous. These millions of deaths have been caused mainly because of the human to human transmission through the respiratory infection, and with the very short incubation period leading to large number of people getting affected at the same time. Bubonic plague. When the Yersinia pestis bacteria enters the body through the bite of an infected flea or through a cut or a break in the skin, resulting in the disease called as the bubonic plague. So Yersinia pestis can enter your body directly through cuts on the skin or through the bite of an infected rat flea, Genoptera chops. There is a sudden fever, chills, prostration, painful lymphadenitis, depending upon the site of the bite. Suppuration, a favorable sign is seen when there is that means that the infection is localized and it is not spreading. Sorry. Most common form of disease, if left untreated, can progress to septicemic plague. The most common form is the bubonic plague, and it will, if it is not treated on time, it can produce septicemia because plague, it's known to produce both exo and endotoxins, this bacilli. Pneumonic plague, usually a complication of bubonic or septicemic plague where the sputum is positive for plague bacilli. Yersinia pestis bacteria are inhaled and lodged in the lungs causing pneumonic plague. It can spread from person to person. So initially the infection which was transmitted by the rat flea from the rodents, and initially it was limited. Pneumonic plague is a dead end disease. Pneumonic plague cannot spread from human to human. And if we take measures to control the rat fleas, the cases would come under control of the bubonic plague. Bubonic plague is a dead end disease because the bacilli remains limited to the buboes or the lymph nodes. But once the septicemia occurs and once the bacteria reaches the lungs, and through the lungs it can come in the cough and the nasal droplet nuclei, and it can then spread from human to human. So in the early part of the infection or the outbreak, the disease is not spreading amongst human to human. But later on, as the more and more cases of pneumonic plague occur, then there is a human to human spread. Septicemic plague, primarily septicemic plague is rare except in the lab infection. Usually it is a complication of bubonic plague. Yersinia pestis accumulate in the bloodstream and they can cause septic shock. Resulting disease is called septicemic plague. Yes. So the Yersinia pestis which accumulates in the bloodstream can cause septicemic shock, resulting in the disease called as the septicemic plague. Lab investigations. Staining with Weissen stain or Giemsa stain. You have to do the staining using Weissen stain. On the stain it shows the safety pin appearance. Then we can culture the secretion of the bubo or the sputum or blood. The transport media which is used is the Clary-Blair media, and you can obtain the convalescent sera and the acute specimen and do the serology. Other methods are inoculation in guinea pig and immunofluorescent microscopic test. Now, how do we achieve the prevention and the control in the case of outbreak? We have to do the control of cases, control of flea, and control the rodents. We have to do three strategies are there: vaccination, chemoprophylaxis, and health education for the cases. So let's see each of this. Now first is the control of cases. EDPT, early diagnosis and prompt treatment. Early diagnosis is through the clinical features. We have to do the early diagnosis: high grade fever, the buboes, chills, and the history of rat fall in and around the house. That gives us the indication. This history is very important, that if the patients or the community reports a lot of rodents dying in and around the vicinity of the houses, then there is an indication that it can be plague. Then prompt treatment you have to give. With streptomycin, it is given 30 mg per kg body weight intramuscular injection for 7 to 10 days, or tetracycline 30 to 40 mg per kg body weight for 7 to 10 days per oral, or penicillin and sulfonamides. It's very important that as soon as the disease is diagnosed or when it is suspected, we have to notify the disease. The disease is under international health regulations, is must to be notified. The patients usually should be isolated. Although bubonic plague does not need isolation, because human to human spread is not possible through bubonic plague. Still, as a precautionary measure, it's better to isolate these cases. Pneumonic plague, even if suspected cases are there, they should be isolated. So that was about the control of the cases. Then control of fleas. Insecticides, most effective as dust, 2 to 3 grams per square meter area. DDT 10% or gamma HCH or dieldrin. BHC 3%. Carbaryl 2%. Malathion 5%. Lysinol 2%. So those are the insecticides that are spread: DDT, BHC, carbaryl, malathion and lysinol. Then the patch dusting on all the walls above 1 feet and burrows. Dust treatment of animals, cat, dogs and pets. So all these animals, vertebrate animals in and around the humans, like the cats and dogs, they are dusted with the using the insecticides like HCH or malathion or DDT. Insecticide spraying up to 5 miles around each infected locality, and within 48 hours of application, the flea index should drop down to 0. Repellent which we can use is diethyl and dimethyl toluamide, DEET. So we have to control the cases, because once the cases are detected, you have to start them on antibiotics, IM antibiotic or oral antibiotic tetracycline. Then you have to take measures for the control of fleas. As soon as we spray the insecticide, the flea index should drop down to 0. Then we have rodent control measures like the sanitary measures, trapping, rodenticide, fumigation and chemosteroid. So there are various measures. We can trap the rodents, use rodenticides, chemosterilization. Sanitation methods. We have to improve the sanitation of the place by proper storage and collection and disposal of garbage. Food stuff should be stored properly. Food should not be made easily available to the rodent. Construction of rat-proof buildings, godowns and warehouses, and the elimination of rat burrows. These are the important measures by which we can keep the area clean, keep the villages or communities clean, and thus improve the sanitation. Trapping. Number of traps should be 5% of the human population. Wonder trap developed by Hafkin Institute can trap at 25 rats at a time. Then we can do the chemosterilization procedure on the rodents so that they don't produce more rodents. WHO expert committee on rodenticide classification. Rodents require the rodenticides requiring ordinary care, red squill, norbromide and zinc phosphide. The requiring maximum precaution is sodium fluoroacetate, fluoracetamide and strychnine. And too dangerous for use is the AS2O3, ANTU and gopacide. Commonly used poisons in India are the BaCO3, barium carbonate and zinc phosphide. Multidose poisons warfarin, diphenas, conferal and pindone. These are the various rodenticides which are available. Now we come to the fourth thing what we can do is vaccination. So the first three things were control in the cases, control of the rat flea, control of the rodents, and now the vaccination. WHO recommends vaccination only for the prevention and not the control of human plague. We can prevent the plague by the use of the vaccine. It should be carried out at least 1 week prior to the anticipated outbreak in 2 doses. So we have to give 2 doses. Vaccine was first discovered by Hafkin. Vaccine currently in use is the Hafkin formalin-killed plague vaccine which was modified by Suki. Yersinia pestis grown in the casein hydrolysate broth for 2 weeks at 27 degrees and then treated with 1% formalin. Now it contains almost 2000 million per killed organisms per ml. The schedule of primary vaccination is 2 doses 17 to 14 days apart. However, in emergency, primary schedule injection and double the second dose can be carried out. Booster doses has to be given after every 6 months. When recommended during epidemic or before travel to endemic countries, subcutaneously administered on the deltoid or the anterolateral aspect of the thigh, and there are no contraindications for its use, and the common reactions are pain, tenderness and headache. Now this is about the Suki vaccine. In adults we have to give 1 ml first dose, 1.5 ml second dose and 1 ml 6 monthly dose. Again in the females 0.75 is the first dose, 1 ml second dose, 0.75 is the booster dose after 6 months. Similarly, reduced doses are given to children. Then what is the chemoprophylaxis? Whenever there is an outbreak of plague, we can give chemoprophylaxis. We can give the antibiotic drugs to people who are not suffering right now. So it is recommended to all the contacts, all the nursing, medical, public health personnel, those who are at the risk of getting exposed to the infection while taking care of the patients. The drug of choice here is again tetracycline, 500 mg 6 hourly for 5 days. Sulfonamide is 2 to 3 gram daily for 5 to 7 days. So tetracycline 500 mg 4 times is easy. Now what is surveillance? So apart from controlling the disease amongst the cases, rodents and fleas, what is important is the surveillance. So What do we mean by the term surveillance? Continuous scrutiny of factors that determine the occurrence and distribution of disease and other health-related conditions or events. It's essential for the effective control and prevention. It includes the collection, analysis, interpretation, and distribution of relevant data for action. This is the definition of surveillance. It's a continuous scrutiny of factors. We have to look out for the various cases, and we have to look out for conditions which are favorable for the occurrence and distribution of disease. It also includes scrutiny of health indices, nutritional status, environmental hazards, health practices, and other factors that may affect the health. Epidemiological surveillance, demographic surveillance, serological surveillance are various examples of surveillance, and the purpose is to detect the changes in the trend of disease in order to initiate investigation or control measures. So why was the need of doing surveillance? This is in order to detect the trends or the different changes which are occurring in the magnitude of the disease and initiate early control measures. Now, responsibility of the surveillance team: detailed study of all the cases and dwellings of humans plague in order to establish the rodent vector source of infection or man-to-man transmission. Issue questionnaires to the population at risk. Isolate the subjects and why best is for detailed biochemical analysis. Serological survey of the target population about clinical cases, recovered infection, and asymptomatic pharyngeal carriers. So we have to do the serological survey of the target population, where we target the subclinical cases, those who have recovered and those who are suffering asymptomatically. Investigate changes in human population, their socio-economic activities. Keep a watch on the containerized cargoes of grain and other food crops originating in areas with natural plague foci and destined for international trade. Undertake surveillance in cooperation with adjacent countries if natural foci are common in 2 or more countries. Give special attention to seaports, airports proximal to natural foci, epidemics or exotic areas. Surveillance of all aspects of ecology, entomology, mammalogy, epidemiology, and preventive measures. So not only you have to keep the surveillance of the rat flea, but also you have to do the surveillance of the ecosystem, the fleas, the rodents, and the human species. We have to give special attention to areas where there is a transit and when there is a border areas, and when the goods and the cargo they arrive from other countries. We have to be very sure that the cargo is not infested with rodents or there are no rat fleas. So special attention need to be given to the cargo, especially those containing the agricultural produce. Health education. Prompt reporting of dead rats and suspected human cases so that preventive measures can be taken. Medical practitioners should keep plague in mind in different diagnosis of any case of fever with lymphadenopathy or when multiple cases of pneumonia occur. Reporting of cases and outbreaks of plague. Local administrative areas, they have to be notified. First, we have to have a map of the area. Then field investigations are done and the types of the plague cases and the numbers are where they are detected and the place where detected. All the data is generated. How many are clinically confirmed cases, how many are lab confirmed, recovered cases, age of the patients, deaths, etc. is noted down. Contacts of the known persons and whether their serological test is done, throat swabs are taken, and Chemotherapy is applied if there are no direct cases seen. Then identification of domestic and wild rodents, identification of Synopsera chirpis and species, resulting in lab tests or infection, results of insecticide resistance, etc.